Welcome to the 785th monthly meeting of EPAASM. Today we have Dr. Karius Test from UPenn, as always. We'll hold all the questions toward the end. If you want to ask your questions in person, virtually, hit the participant tab and hit the blue raise hand button so I can unmute you and you can turn on the video and you can ask your questions. Alternatively, you can type your questions in the chat box and I'll relay the question. So please join ASM. We provide a community and network as well as career development opportunities. As well, a lot of great science, either in a meeting or webinars or virtual posters. And there are a lot of resources online, keynote lectures, open sessions. And we would, of course, encourage you to join the Eastern Pennsylvania branch when you sign up for ASM to support all the great lectures and activities of the branch. So today, we have Dr. Kyle Rodino. He's assistant professor in clinical pathology and laboratory medicine at UPenn. He's also the assistant director of clinical microbiology laboratory in the hospital of the University of Pennsylvania. He is a very new addition to the Pennsylvania microbiology community. He just arrived in last August. He's gotten his PhD from the Virginia Commonwealth University. And then he was a fellow at the Mayo Clinic at Rochester, Minnesota. He's board certified for the American Society of Clinical Pathology as well as the medical microbiology. He's had a long-standing interest in using new technologies to diagnosis as well as detection of infectious disease. And today he's going to tell us about the next-gen sequencing in clinical microbiology, the current applications, and what to say.
Thanks so much. While I share my screen here, I just want to say thanks to Vincent and thanks to Irv, who actually invited me to give this talk. I've heard a lot of great things about this branch, its long history, and also its very active membership. So I'm really happy to be talking with you today. I have no disclosures, and my objectives for this evening's talk are first to very quickly define next-generation sequencing and review some of the current applications that have made their way into clinical microbiology. The bulk of the talk is going to discuss the use of metagenomic next-generation sequencing for pathogen detection, some of the challenges that are faced while implementing that in a clinical space, and then also the challenges that even if you don't implement it in your laboratory for routine use. And then I'll end with some potential applications for NGS and clinical microbiology, some of the things that I think are coming down the pipeline or would be interesting to look at. So next-gen sequencing goes by many names, high throughput sequencing, deep sequencing, but the one that I think is most accurate and describes the method is massively parallel sequencing. And this encompasses a number of platforms, so-called second-generation sequencing or short-read sequencing, which is dominated by Illumina, but also includes Ion Torrent and some others. These generate reads that are relatively small, about 300 base pairs or so or less. Their advantage is that they have very low error rates, but assembling these really small fragments can be difficult. The next set of platforms are so-called third-generation sequencing, or if you look in some other places, even fourth-generation sequencing, is long-read sequencing, which encompasses things like PacBio and Oxford Nanopore technologies. And these generate comparatively much larger reads, 5,000 to 100,000 base pairs, maybe even into the millions. They're notoriously prone to higher error rates. However, because they produce these really long reads, they're easier to assemble genomes, etc. The really big difference between next-gen sequencing and Sanger is, of course, the volume. So in this very simple depiction of what's going on, we're sequencing by Sanger, nucleotide by nucleotide, and getting this a very small amount of information. And if we talk about Illumina, this sort of is happening in parallel, but it's happening on such a massive, massive scale that really the advantage to next-gen sequencing is just the amount of information that you can obtain. When we look at a next-generation sequencing workflow, it's really sequence and then assemble, right? So you can start with either purified organism or initial clinical sample that may be full of many organisms, extract that nucleic acid, do some library preparation, get that material onto whichever sequencing platform that you're using, short read or long read, using a complex network of bioinformatics and pipelines. You then assemble those things back into full genomes, and you can detect the organisms that are there. You can differentiate them. You can do a whole number of downstream applications. To cover some of the applications for NGS currently in the clinical space, I wanted to highlight a few things. So first is organism identification, and NGS really allows for increased resolution of some hard to differentiate organisms. And here's just an example of that. The Mycobacterium abscessus complex, which is made up of 3 subspecies, can be very challenging to tell apart using routine methods, but using NGS, we have a much better resolution and the ability to tease these subspecies apart from each other. NGS has also made its way into outbreak investigation. We know that that level of information that it provides is much more discerning and provides greater precision for pulse field gel compared to pulse field gel electrophoresis. The whole genome sequencing applications for things like Listeria, E. coli, Salmonella are now commonplace in the CDC for foodborne disease outbreaks, and PulseNet has essentially been replaced by whole genome sequencing net at this point. But we've also seen NGS move into the clinical microbiology lab and say the investigations for hospital infection control, where you're looking for linking spread of organisms around a unit and saying whether or not they're related. And then finally, I think one of the methods that has popped up in the last few years is the genotyping and resistance determinantability of next-gen sequencing. So with the increased sensitivity for detecting mutations compared to Sanger, we've seen the ability to detect viral, specifically resistance, at a much earlier rate than previously. So this application includes things like HIV and CMV, where the resolution of next-gen sequencing allows for detection of much, much smaller populations of virus that possess a mutation. And then in the example here, I'm showing CMV, where there are resistance determinants that are well mapped and good databases for comparison. And we can look across regions of the genome, specifically UL54 or UL97, and find those mutations that map to some sort of viral resistance. But really what I want to talk about today, this evening, is using next-gen sequencing for pathogen detection direct from sample. And this is a really nice depiction of really the method and how powerful it can be. So you can essentially take anything, put it into your workflow, and then ultimately out the other end comes detection of pathogens from that sample. When we talk about detecting pathogens with NGS, I think there's 2 main ways that that's really done. So targeted next-gen sequencing, which is primarily amplicon-based, where you're looking for a specific gene that's shared amongst a group of organisms. So say the 16S rRNA gene for bacterial detection. And I think some people refer to this as metagenomics, but really it's more specifically referred to as metagenetics, where you're looking at a single gene and not the entire genome. But the idea is really that you take your clinical sample, you have bacterial nucleic acid and also host nucleic acid that's extracted, but then using those specific primers for, say, bacteria, you amplify only the bacterial pathogens, and then do library preparation, sequence them, and ultimately identify the organisms that are present in the sample. The advantages of this is that it alleviates that burden of human DNA in that amplification step, you also get an improved sensitivity of the organisms that you're looking for with a less depth of sequence because you've cut out so much extraneous material. But this does still require some sort of suspicion for at least a particular class of organism when you're setting up a test like this. The other way to do this is with the unbiased approach, or shotgun sequencing, where you have metagenomic next generation sequencing. And for the rest of the talk, I'm just going to call this metagenomics. So the idea here is that you're assessing all of the genetic material, DNA and or RNA from a sample. And this is really what I think people would consider the holy grail of pathogen detection, because it's entirely agnostic and hypothesis free. You do not have to have a suspicion of a particular organism. The test does all of that for you. So the idea here is that you have DNA, RNA, again, host nucleic acid, and then contaminating nucleic acid. And all of that is library prepped, sequencing occurs, and then through the data analysis part, you eliminate the things you don't want and report the things that you are interested in. The drawbacks here is that it is highly susceptible to contamination. And then everything relies on that reporting step and the analysis step in the software, so you get a complex data analysis and reporting algorithm. When talking about at least the theoretical advantages to metagenomics, I think it's nice to look at this slide where we can look at the traditional workflow compared to that metagenomic workflow. So on the traditional side, we essentially have all of these different sample types that get processed and plated differently depending on what type of organism we're looking for. So you can process and plate differently and have different workflows for parasites versus bacteria, etc. The workflows are measured for the routine things usually in days, up to a week or so, but we know that some of our slower growing organisms like mycobacteria and fungi, the workflows are measured on the order of weeks. And then of course we have these newer rapid molecular testing that have come through in the last few years, where we can look for some of these traditionally slower growing organisms with these targeted methods in a much shorter time frame. Sometimes on the order of days, but in the example of syndromic panels, maybe on the order of hours. When we look at metagenomics, some of the advantages are, in theory, you can take a sample and put it in the pipeline, and it's handled exactly the same way, no matter the type of organism that you're looking for. The timeline is, in theory, a couple days. It would take you 2 days or so to get through this entire process. I think that the timelines on the left are more realistic, daily. The lab can manage these timelines continually. And the timeline on the right, there are only maybe a few laboratories or clinical laboratories in the world, I think, that could really manage a 2-day timeline continually doing the testing. But this is a possibility. And then the last thing I think really speaks to the advantage of NGS and metagenomics, and that you get so much information out of this. So not only do you get identification, but you could get things like resistance determinants, you could get host response if you wanted it. You get so much information, and it really encompasses all the information that you could get out of some of the traditional workflows in one test. With all that said, there are a number of hurdles to metagenomics in the clinical lab. And I broke them down into challenges of implementation and then challenges of use. So when we talk about challenges of implementation, I think the first is assay optimization. So what we know is that every step of this pathway that I showed earlier impacts the results. And you have to look at them all individually to see what the cumulative impact on the outcome and the results can be. So for assay optimization, I think the things that we consider are the decisions around nucleic acid extraction. Are you going to do some sort of pre-processing step for your sample? What sort of efficiency is there for different sample types? You're going to have to make decisions around amplification, enrichment of organisms, depletion of host reads, and are you going to look at DNA, RNA, or both? And then library preparation and sequencing. And this really makes up the wet work of what you consider for implementation, essentially all the things that are done on the bench. And then next we'll talk about things that are done on the computer or the dry work. A few years ago, Trish Simner at Johns Hopkins published a really nice article outlining some of these challenges of the assay optimization steps. So she was looking at metagenomics for spinal fluid, and they kept things the same, like the extraction platform that was used and the analysis pipeline, or a very similar analysis pipeline. But what they assessed was pre-processing. So was the spinal fluid centrifuged? Was there a bead beating step in the pre-processing portion? They also looked at using DNA or RNA as the starting material for their sequencing, and they looked at a variety of organisms that could be encountered in spinal fluid. And really what they were trying to see is, as we make changes to the workflow, how does that impact the results? And what they found was absolutely what we would expect, I think, is that sample processing greatly impacts the sensitivity of the test, and that impact can be organism specific. So as a few examples on the right here, for Pseudomonas, what they saw essentially was that for no matter what fraction of the spinal fluid you looked at, the pellet, the whole sample, or the supernatant, the absence of bead beating and then using RNA as your starting material and converting it to cDNA was the most sensitive for this organism. But when you look at something like Cryptococcus neoformans, it was really dependent on the fraction of the spinal fluid. So for the pellet, there was great sensitivity no matter the processing steps that you used. But if you looked at the supernatant, there was really poor sensitivity no matter what processing steps that you used. So the way you set up and work through the analysis really impacts the results. The other thing that this study showed was that metagenomics can be less sensitive than a targeted test. So for example here, the Cryptococcus neoformans in a low concentration was positive by routine testing, but was negative by no matter what method you used when looked at through metagenomics. So we know that the setup and the wet work part is impacts the results. We also know that adding extra manipulation steps may increase the background contamination. So you need to balance sort of how many steps do you want to put into this before you start doing the sequencing. When we transition over to dry work, or the stuff essentially done in the computer after the benchtop work, you have to consider the analysis pipeline, the database you're using to generate taxonomic calls, and then how you're going to report that. I think the challenges to the dry work steps are probably some of the most challenging for traditional microbiologists. It's really not something that we have regularly had to do, and the complexity of the software needs should not be understated if you're building this pipeline and analysis from scratch. So in order to process those sequencing reads, you need to have software that can do the removal of poor quality reads or known errors that exist on your sequencing platform. You need to have a system to subtract the host reads and look at only the microbial reads. You need to have software for alignment and taxonomic classification. And in most cases, this is not just one piece of software, right? This is a group of programs that work together to accomplish all of these goals. For the last part, the taxonomic classification, what's really important are quality curated databases. Databases that are updated to include new organisms or new strains, and also databases that have been parsed through to remove any of the inaccurate classifications. So just to show you a really simplified example of how important the database can be, this is from some work that I was doing. This is actually a targeted next-gen sequencing assay, but it will get the point across in a simplified manner. We started with whole blood that we knew contained Ehrlichia murus oclarensis, and we subjected that to next-gen sequencing. And on initial data analysis, we had a large number of the reads corresponding to something that was called result below the cutoff, and it gave a poor ID percentage. So what we actually did is went through a database update, and we found that Ehrlichia murus oclarensis was not in the database that we were using. But with that update, it was now included. And you can see the same number of reads are now mapped to Ehrlichia murus. And when we look at exactly what this is, the type strain for Ehrlichia murus oclarensis. So this is just a very simple way to illustrate that the database really dictates your result. I think it's also easy to imagine that if this organism hadn't been missing, and say there was a misclassification of the organism, we would have had no way to know that. and misclassifications can be really difficult to detect and end up with you reporting the wrong organism. Once you've established which software you need and what your pipeline looks like to accomplish your goals, remember that often these softwares are not, they're not with the clinical needs in mind. So from the regulatory standpoint, you need to have a way to deal with version control and making sure that you're locking down the database and the software that you're using so you don't have updated versions where you're unaware of the impact on your total pipeline. With all the data that's being generated, we need the ability to have secure storage. It's again uncommon for the clinical microbiology lab to be dealing in terabyte-sized files of information. And when it comes off of the sequencing platform, it certainly contains a large amount of patient information, which are protected under the HIPAA requirements. So the storage and the security are really important when dealing with metagenomic data. And then lastly, once you have the data processed, you need to figure out exactly how you want to interpret and report that. So some of the challenges that go along with this is establishing the clinical significance or understanding what could be assay background. And what we know is that metagenomics, one of its advantages is its ability to identify very rare or novel pathogens in a clinical sample, something that you may not expect to see, something that may be hard to reconcile with the clinical picture. So do you have some sort of process in place to confirm the results, say sending to the CDC for a targeted assay? And something that's been used in a few models is having an expert review panel, something similar to a tumor board, to look at the patient's clinical history, the result, put them together and decide if we feel as though that's an actual believable result. But ultimately, there's also going to come down to the responsibility to sort through suspected contaminants. So we know that DNA and RNA are absolutely everywhere. They're in the laboratory. They're in the kits and the reagents that are part of the assay. They're in the collection devices that the sample comes in. So knowing the assay background, understanding what is there and what can and cannot be reported and what may be an artifact of the actual test is really important for the person running it. And also to appreciate that background contamination may shift as time changes. When the lab changes, when products change, you may see shifts and new things appear in that assay background. So it needs to be continually addressed and readdressed. All of that just encompasses the optimization portion. And the other part that should be stated is that this can be really expensive. So sorting out all of the options and going through a full clinical verification of an assay like this can cost north of $100,000 per sample type. So really something expensive if your lab is considering bringing it in-house. Once you have a test that works, there's still other challenges, right? So now you have to decide, how does the test fit into the routine workflow of the laboratory? What sort of people and skills are you going to have to run this on a routine basis? What is the expected volume that you're going to get for testing? And then once you're established sort of the demand for that test, that's really going to impact the potential throughput. So are you batching samples weekly and the results are going to be coming out weekly? Are you expected to be running it every day? And do you have enough samples to be running it every day? And all of those considerations really play into the turnaround time for a test like this, where you have to be efficient and also be responsible with the resources. How often should you be running it? And then if you're running it in that time frame, is it clinically actionable and providing results in a time frame that people really need? And then the last thing is a discussion about reimbursement. So if anything, the history has shown us that the syndromic panels that came out initially started getting larger and larger, and then the reimbursement around those was shaky, and it's not so easy to get a full 13 pathogen panel reimbursed at this point. And I think that it's unclear what the reimbursement climate is going to be like for these metagenomic assays that essentially can detect anything. So spending a lot of money and developing a test like this and putting it in place with an unclear reimbursement climate, it may be challenging. There have been some recent improvements in bringing these and guidance how to bring them into a clinical space. So very recently, there have been a number of published roadmaps of actual clinical verifications, and they've shared the pipelines in which they put together to develop these assays. There have been a number of groups that have been working on suggested quality metrics, including the Next-Gen Sequencing Working Group, which has a number of publications that have been out in the last few years. There are CLSI guidance documents, and there are numerous publications that discuss the quality thresholds and reporting thresholds, regions across the genome that are how many regions you should see in order to determine that something actually came from the sample and is a real detection. Some advice on what to use and how to use internal and external controls as quality metrics. There have also been recently some both public and commercial curated databases that are designed for clinical use that have gone through that vetting process of eliminating the maybe misclassified or erroneous reads, but also have some scheduled updates that allow versioning to be easier and also make sure that you have updated organisms being included. And then finally, just as we saw syndromic panels emerge, there are sort of next-gen sequencing black boxes that are coming out that are, for many labs, are a much more approachable platform. So some of those, for example, are Illumina is coming out with a kit-based sequencing platform that connects with a commercial data analysis platform at the Explify platform by ID by DNA, specifically looking for respiratory pathogens. There's the Ion Torrent Genexus system that's distributed through Thermo, which is essentially a sample, or could be a sample to answer system for next-gen sequencing. Of course, the trade-offs are the same as we've seen with those syndromic panels, where you have a loss of flexibility, a loss of customization. You have a limited ability to evaluate what's going on in the entire process. And then ultimately, for the lab standpoint and the director standpoint, a loss of control, where you just essentially have sample going in, report coming out, and you're not really able to evaluate much beyond that. With all of those challenges, I think that implementing metagenomics in particular is challenging for a routine clinical micro lab. But there have been a number of laboratories that have gone through this, and they're offering those tests from the reference lab standpoint. So the next part I wanted to talk about is what are the challenges in just using metagenomics in practice. And there's three really metagenomics tests that have come to the clinical space: CSF, plasma, and respiratory samples. And I think there's the most data around the CSF and plasma, so I'm going to talk about those two in particular. So for CSF metagenomics, this is pathogen detection direct from spinal fluid. This was originally validated for clinical use at the UCSF Center for Next-Gen Precision Diagnostics. It's a test that looks for both DNA pathogens and also RNA viruses, and it does utilize a human DNA removal to enrich for these organisms. And if you're interested in the details of how they went through this laboratory validation and a really great roadmap on how to do this, this is a great article for Clin Micro folks to understand how they went through that validation, then ultimately chose reporting thresholds and other considerations. The test has been used and shown to detect bacteria, mycobacteria, viruses, fungi, and parasites, and there are numerous case reports showing the success of metagenomics from spinal fluid. I just wanted to highlight a couple of them. The first one that I can remember seeing, I think maybe the first published case report, was from 2014, the New England Journal. This is a 14-year-old SCID patient who had 4 months of fever and headache. Using the metagenomics assay, they detected a very small number of Leptospira reads, and this was actually confirmed by a CDC assay. The child received targeted therapy for Leptospira and ultimately recovered. So I think this is a great example of how all the prior testing was negative, and then metagenomics really provided some information that wasn't there and impacted patient care. The next is an example of how metagenomics can be used to detect those exceedingly rare or novel organisms. So this is a 34-year-old from Australia who had XLA, and the patient had 3 years of undiagnosed meningoencephalitis. Ultimately, using metagenomics, they detected Cache Valley virus, and this was confirmed by brain biopsy PCR and also immunohistochemistry. Interestingly, in Australia, there was no known Cache Valley virus, and this patient actually traveled to the US some small interval before he began experiencing symptoms, and it was thought that he contracted this while in the United States. So one of the things that was tried was this prompted treatment change from IVIG in Australia to IVIG in the US in an attempt to find antibodies that may help fight this. Beyond the case reports, there is one multi-center prospective study that was published in the New England Journal in 2019. This was a study that looked at 204 patients that were admitted for meningitis or encephalitis. Using a multitude of diagnostics, including routine methods and next-gen sequencing, 58 infections were diagnosed in this group of 204 people. For the diagnoses that occurred by next-gen sequencing only, they detected a number of organisms, Candida tropicalis, EBV, echoviruses. Ultimately, 7 of the 13 infections that were diagnosed by metagenomics only actually impacted treatment. And we look at the total of cases, 204, and metagenomics alone diagnosed, if you want to say only the cases that impacted treatment, about 4%. If you want to say at any time a organism was identified, about 7% of cases. So a small impact on the overall number of patients that were admitted. When you look at the diagnosis that occurred through routine methods only, which was 26 of the 58 infections, serology made up the bulk of this. So serology actually diagnosed 11 of these infections. There were other clinical samples that were involved in the diagnosis of 7. And then there were 8 that were made from the CSF using routine methods. And the thought here was that metagenomics missed these organisms because they were at very low titer in spinal fluid. The summary of this prospective study is really, I think, that in some cases, it is undeniable that metagenomics provides diagnosis where other methods fail. However, other methodologies or samples may outperform metagenomics depending on the clinical situation. And I think it showed very easily that metagenomics does not replace routine testing. However, it also raised a few questions. And I think one was that the cohort that they studied was really well defined, right? These were essentially all acute infections. They were all admitted patients. They all had meningitis, encephalitis, and it was really well controlled. And we still saw what I would say was a small increase or benefit to adding this testing. But the question remained that, is this really a real life application of the test and a real life expectation on the return of the results? And can further studies help define clinical utility? So when I was a fellow at Mayo Clinic, we actually reviewed all of our send out metagenomic testing. So this encompassed 80 samples that were sent to UCSF for testing. At Mayo, we had no barrier to ordering. There was no approval process or something that people had to go through in order to order the test. We found that the majority of the tests that we sent were actually used for subacute or chronic conditions, not the acute conditions that were shown in the New England Journal article. The ordering was equally split between inpatients and outpatients. About 50% of the tests that were ordered were for rule out of infection prior to starting an immunomodulatory treatment. So a scenario where both infection and an immune process were on the table. The ordering services that we saw most commonly were 1, neurology, and 2, infectious diseases. And then almost exclusively, this test was ordered after extensive and really exhaustive routine testing. What we saw in this review is that of the 80 samples we sent, we received 20 positive results. 5 of those results corresponded to potential pathogens. 2 of them presumably could have been detected by methods other than metagenomics, but because some of the samples that we were looking at actually came through as reference lab samples, we didn't have access to those routine methods. So those included Toxo and West Nile virus, but we don't know if they were detected by other methods. And then really interestingly, 7 of the 12 positive results were thought to represent likely contaminants. So organisms that were not known to cause clinical disease, were connected with gut flora or et cetera, or had comments about having other organisms from gut flora just below the threshold of reporting, something like that. The other thing we noticed, and because it seemed like a lot of providers were ordering this test as like a final rule out of infection, as we looked at the negative results. And it was interesting that 10 of those had a comment associated with them reporting that there was potentially reduced sensitivity of, say, DNA organisms or RNA organisms. This wasn't really a new finding, though. In that New England Journal article from 2019, it was shown that in samples that had higher cell counts, that there was actually an increase in the DNA or RNA background that was detected, presumably because there was so much human nucleic acid in the sample that actually impacted the sensitivity of the test. And then one final observation that we had from this study was that often the second or third lumbar puncture was needed because the initial sample was exhausted. And knowing that for some organisms, say arboviruses, have a very, very short presence in spinal fluid, the question came is that maybe the sample that we're sending really is that no longer has that causative agent in it by the time metagenomics comes up. But I think that the idea of reserving sample is really more challenging than it appears, right? So there's some operational hurdles. One, are you reserving sample for a possible metagenomics test and then limiting sample that goes towards maybe more valuable routine testing? In some cases, you don't know that you're even considering metagenomics initially, and it's not until later that that conversation comes up. And then just from the lab standpoint, what does storage look like, and how do you operationalize storage of sample for so many lumbar punctures for the potential to maybe send some metagenomic testing? So a hurdle that really hasn't been addressed yet. Moving towards plasma metagenomics. So this is pathogen detection direct from plasma. I think it's commonly now referred to in the literature that's published as the Karius Test. This is a commercial assay. This is looking at cell-free DNA only. It's billed as a much more non-invasive test because it just requires a blood draw, and it is akin to a liquid biopsy where you could essentially look for infection or infection monitoring in severely immunocompromised patients like post-transplant HIV, HEMONC. And I would say that I see it being increasingly common in pediatric centers where the thought is that it's easy to collect in a cohort where maybe there's not such good diagnostic hints and diagnosis can be challenging. But there are no large prospective studies, only retrospective reviews that we're going to talk about in a little bit to guide its use. There are, similar to CSF metagenomics, a number of case reports for pathogen detection using cell-free DNA metagenomics, a list of organisms here just as examples. But again, what is the experience with routine use is what the real question comes to when considering using plasma metagenomics. So I just put together a table to summarize 4 of, I would say, the largest studies that are out there. The first study, being the largest, was done in conjunction with the company and published in 2019. And then subsequently, we've seen 3 reviews of experience come out in a variety of journals. The first thing to notice is that unlike spinal fluid metagenomics, with plasma metagenomics, you get a lot of positives. And in some cases, you see a good percentage of the results have multiple positives. I think that when you consider the fact that this is commonly applied to a confusing and complicated patient population of immunocompromised folks, reconciling all of these positives can be quite a large hurdle. All of the studies looked to compare the metagenomics approach to some routine methods, usually a composite of a number of routine methods, and you can see that the positive predictive value was variable, and then not all, but most of the studies looked at the negative predictive value, and I think predictably lower, seeing as that we're recovering so many positives that the metagenomics test has a higher reporting of positives than the routine testing. All of the studies looked at the impact of the testing a little bit different, so I just wanted to highlight a few of them. In the first study, they didn't look at this as well, but one thing that I found really interesting is they included a group of asymptomatic donors of plasma, and about a quarter of those people had one or more organisms identified. So they have no known pathogenic process occurring, but you're still getting a positive result and a pretty good clip in this group. In the Hogan study, which I think looked at the impact of this testing best, they divided results that were positively impactful, divided results that were negatively impactful, where some change or management decision was made that actually wasn't in the best interest of the patient. And then there was no impact in almost 90% of the tests that were sent. The last two studies did not differentiate impact based on positive or negative. What they essentially said was there impact in the sense that a change was made in the management of the patient, and they defined this as positive impact. And then no impact was as it's described. So no change was made in the patient based on the result. So something like 10 to 25% of patients had a change. Of course, whether or not that change was good or helpful is unclear. And then again, a much larger percentage, 75% to almost 90%, there was no impact. So I think you can see that there's no impact in the management of a patient much more commonly, even though there's a large number of results returning with positive for organisms. The last thing that they looked at was the turnaround time, and they looked at it a little bit differently, but this test is rather quick, so something like 3 to 4 days from sending to receipt of results, and the Lee study looked at just the turnaround time from when the commercial lab receives the sample, and it was about 1 day from receipt at the lab to resulting. A summary for plasma metagenomics is really that I think in sending you should expect a positive result, and in some cases many positive results. It was noted in these reviews, and I think clear I hope clear from what I've talked about, that the clinical specificity of the test can be an issue. And again, in complex or severely immunocompromised patients where anything could be a pathogen or anything could potentially be causing an infection, I think parsing through that list of organisms may be really, really challenging. The positive impact in the reviews that are coming out is not really clearly defined, and I think everyone's looking at that a little bit differently, but I think the reviews suggest that there's limited positive impact from plasma metagenomics. We definitely need more studies to determine the true utility of this test. And then one of the things that I didn't talk about, but that I think it would be important, is to look at the reporting units. So these results come with something called microbial cfDNA per microliter, or MPM. And the Lee study, they tried to look at if a higher MPM correlated with it being a more significant organism, but I think how to use this unit that comes with the report still is up in the air. As a full summary of the challenges of using metagenomics clinically, I think the biggest thing is that we have a lack of guidance on clinical use. So what patients and when is this likely to be most helpful? We know that in order to generate that data, there's probably a considerable amount of overuse going on, and this is a great time to talk about cost. So certainly when metagenomics impacts a patient positively, the $1,000 or $2,000 it costs for this test is really nothing. And also that information that's generated, if it can prevent additional days in the hospital or additional testing, the cost of that test is really minor compared to what some of the cumulative cost could be. However, if we're using the test frequently and overusing the test in cases where it's unlikely to be helpful, I think that greatly increases the cost per actionable result. And that's a burden that the health system has to absorb when we're sending 100 tests and getting 1 or 2 back that are actually informative. It really becomes an expensive test when you think about it in the global standpoint. We know that because of the limited guidance and we're using it in a lot of patients, it's not going to be valuable, we're increasing the likelihood of unexpected and confounding results. And then ultimately that falls on the laboratory and the clinicians to look at them and provide some sort of expert review, which can be really challenging. I think from some of the studies that we looked at originally from Johns Hopkins, and then also with the prospective and retrospective reviews of CSF metagenomics, is that there's a false perception that the test is ultra sensitive. And the reality is that just like every test that's looking to detect an organism, you can only detect things that are present in the sample, and it brings about that question of optimal sample selection and sending the best sample for the greatest likelihood of a good result. We also know that background contamination and in some cases excessive human DNA may reduce the sensitivity of the testing, and then also that other methods or sample types may outperform metagenomics in certain clinical scenarios. And the last thing that's a challenge is finding valuable results. So one, this can be termed in turnaround time, right? So if you're sending a test that takes a week or 2 weeks and someone is acutely ill, getting that result a week and a half later may have limited value. But the other thing that we should talk about, I think, is actionable versus informative results. So actionable results, I think, being things that you have a treatment for and you can act on the result that comes back. But informative results, I think we've seen this in CSF metagenomics sometimes, is that you're detecting exceedingly rare things that don't have a treatment, but that information is ultimately valuable, right? It can prevent additional testing. It could give patients and families answers, even if it comes back after someone passes. That information that is generated is potentially valuable, even though it may not be actionable. With the last few minutes, I just want to talk about some of the future applications that I think are really interesting and potentially could be of great benefit. So the first is that I think some of the things that are next in next-gen sequencing are really rapid sequencing, where we can provide quick, cheap, essentially individualized sequencing. And some of this is being worked on platforms like the Oxford Nanopore technologies, where they have this flongle that is a couple $100 and essentially could give information in a few hours. What comes along with this is the need for increased computing power and databases to have real-time streaming data analysis. So essentially the results are available as the sequence is generated. And I think one of the really interesting applications for this would be in the detection of pathogens directly from blood. So the study that I'm showing here was actually a study where they used Oxford Nanopore, but this is direct from blood culture, so there's an enrichment. But what you can see is that sequence generated within the first 10 minutes started identifying the organism, and then within an hour or so, they were able to start mapping to resistance genes that could predict the susceptibility of some of these organisms. So with improvements on this in this area, I think next-gen sequencing could really impact say blood culture diagnostics and a number of other things where speed is really an important and someplace we can gain. One of the other things that I think is really interesting is that amount of information that is generated about an organism can really allow for additional improvements, including antimicrobial susceptibility prediction. So we can look to provide full antimicrobial susceptibility testing profiles from whole genome sequencing. And what I mean by that is going from a genotype to a phenotype in a report that is fully understandable by a clinical team. And to get there, we're going to need much better databases and things to compare these genotypes to in order to predict that susceptibility testing. But folks have already started working on that, where they're looking at genome-based prediction of resistance and using machine learning to do the interpretation or the prediction of that drug resistance based on the sequencing data. So again, I think there's a lot to come here, but it's really exciting with how fast and how accurately we could potentially be predicting susceptibility. And the last thing that I think is really interesting is the human disease biomarkers. So for a really long time, we've been viewing the human reads as garbage, right? As we're searching for like this organism in a haystack, but really we should be looking at the 99% of the reads that are human. Because maybe they can tell us a little bit about what is going on in that person. And the first time I saw this idea pitched, it was at the ASM national meeting in New Orleans, where Christopher Woods from Duke was talking about this example here, where they were using PCR to determine if a person had a bacterial pneumonia, a viral pneumonia, no infection, or a co-infection, and they were classifying these people based solely on their host response. And about a couple years ago, there was a metagenomics approach to this where it combined both unbiased microbe detection and the host response as a rule out for lower respiratory tract infection in the ICU. And it did a really nice job of predicting if people had lower respiratory tract infection based on these two parts of metagenomics, both organism detection and some host factors. So I think this is also a really interesting option forward where we can use, the majority of a sample is actually human read, and we should maybe not be trying to get rid of it so quickly. So in summary, next-gen sequencing in the clinical space, I think it certainly has the ability to provide a diagnosis where other methods cannot. I think it's very unlikely that it replaces routine testing as a whole. I think that we should know going into it that the results can be confusing and they may in some cases require expert evaluation. We definitely need more studies on the clinical utility and how to guide usage of these tests. They're expensive, they're time consuming, and I think in a lot of cases they may not be useful, but it would be really helpful to be able to define cases where it could be. Some of the questions that I think we need to ask ourselves when we consider next-gen sequencing, either developing it or using it, is what info do I need, and do I really need it? What info, and can that info be provided by next-gen sequencing? Is it really going to give me the information that I want? And then will that information, if I have it, ultimately be useful? Is it easily interpretable? Is it actionable? Do I get it in a time frame that allows me to do what I want to do? And the last thing is that even though I just spent almost an hour telling you about some of the possible advantages of next-gen sequencing, is that other methods are not waiting for next-gen sequencing to come to market, right? There are other things that are in development as well. And as we work out some of these challenges and hurdles of next-gen sequencing, I think these methods are quickly catching up. So things like CRISPR-Cas, which I think would be a great topic for a future talk, may appear in the diagnostic space before we work out all of the details of next generation sequencing. So will this be quickly replaced by something else? With that, I think that's it. I'd be happy to take any questions at this point about metagenomics, next-gen sequencing, etc.
Thank you very much for a wonderful talk. I'm going to start with a couple questions. So you mentioned that, of course, the wet work can really impact the outcome of sequencing. So with the unknown sample, how do you make that decision? I mean, you were saying that you want to use the human reads, and of course, you're going to have to use the RNA. But how are you going to bifurcate what kind of prep work for the wet work for the sequence?
Are you asking how the wet work impacts the result, I guess? And
I mean, it does impact the result, right? So with unknown samples, how are you going to make a decision of what kind of
with unknown samples? Yeah, I mean, I think that's part of the clinical verification, right? You have to create essentially those scenarios where, say, you're blinded to the results, you're using node samples, so you're evaluating what the impact is by the workflow changes that you're making. But that's the details you need to work out before you get in the realm of testing where you really don't know, right? So we need to define, what is my loss of sensitivity if I make this change? What is my increase in sensitivity if I make that change? And then you can do that across a variety of classes of organisms. I think the approaches that have been taken in the clinical space so far have been to pick an organism for each class that you intend to detect, and then do a lot of testing with those. And then secondary to that, run through a bunch of sequencing to verify your pipeline to include more organisms. But it's sort of an ongoing verification and an expectation of, given what we've tried, this is how we expect the wet work to impact the sensitivity or specificity in certain ways.
And the second question is, in a sample of metagenomic data, if there's a novel determinant or island that hasn't been in the database, if it can't align to a known classification or known database, would that get tossed, or is that going to be still useful?
Yeah, I think that's a challenge to work through, right? So if you're using a well-curated database, presumably you're going to encounter something that doesn't match eventually, right? And so I think the options are then to take that sequence or that what is mapped and look at the less curated databases, and then use that expertise to determine, is this something that's absent from our clinically relevant database and is truly a detection that is believable? Or is it probably some sort of artifact? I think that depends on the quality of what you're comparing it to. But those are definitely the challenges that I don't particularly have answers to, right? It's something that could appear, and it will be exceedingly difficult from the clinical perspective to assign importance to something that is very, very rare.
From Toby Eichenstein, she asked, why hasn't anyone used urine as a sample?
Used urine as a sample? I think that there are actually a few groups, and I know that there are research assays that have been using urine as an example. So, for example, there's definitely improved sensitivity for some of those arboviruses for urine as far as the longer detection. And there are a number, I think, of commercial, or at least one commercial laboratory that's offering metagenomics from urine. I think the need in the clinical space and the relevance of the results are the parts that are currently hindering its adoption, right? So it's one thing to create a test, it's another thing to create a use case, and then a third thing to create a report that is helpful and actionable above what is normally provided as a routine test.
From here, for plasma mNGS, it's an ultracentrifugation step used before DNA RNA isolation. I am thinking about bacterial sepsis with low intermittent bacteria releasing blood, but possibly detectable bacterial extracellular vesicle DNA RNA.
Yeah, I don't know if there's an ultracentrifugation step in the described clinical test. I know that on the research side, those additional steps to essentially concentrate the detectable nucleic acid have been applied, and that some of those do work. There's also just the fact that some organisms are not present or don't have a signature present in that fraction. And no matter how many sort of enrichment methods that you use, it's unclear if the same process will work for a broad class of organisms.
Deborah Powell, I have seen results of sequencing of nodular fluid from prosthetic joints that have listed more than 5 organisms, most of which are cutaneous for image interpretation.
Yeah, I mean, this is a perfect example, right? So every step from pre-analytical to post-analytical can impact these results. So if the sample was collected in a container that was sterile, but maybe not nucleic acid free, it could be detected. If the collection of the sample was somehow contaminated, maybe that those organisms were at a really low burden, and in traditional culture, they would not have grown, and this complex situation wouldn't exist. But their signature and their genetic material may be there. Harnessing the power, I think, of metagenomics is one of the parts that we need to work through. What is a clinically relevant detection? How much organism do you need to see in order to actually act on something? But I absolutely appreciate the difficulty that creates.
Our Abbott, are there similar efforts to create massively parallel virology-based assays to fill in the gaps in NGS methods regarding sensitivity loss window of opportunities for detection of pathogens?
Yeah, I think that there are. So there's some really high sensitivity serologic assays that are being developed. I think, again, a lot of this work is coming out of UCSF, and I think they've done a great job in this area. So they published a paper a year or two ago on this high sensitivity serology assay that identified a lot of enterovirus D68 and connected it to acute flaccid myelitis in kids. So I think that that's a really good opportunity to acknowledge that detecting the organism may not be the optimal strategy, that detecting a prior or current presence of an organism with another method that is also improved is certainly a possibility.
Guy touched on commented that at the end you use definition.
Yeah, and some of those tumor board groups that have been utilized to sort of adjudicate the really complicated cases are made up of expert clinicians and laboratorians and scientists working together, obviously, to all give their opinion on what factors into a clinically relevant result. And I would say that ultimately also you can generate as much data as you want to, but if you don't present it in a way that people can understand, it's probably a futile effort.
And great talk, Kyle. And I think we can all agree to that. And thank you very much for giving the talk, and welcome to Philadelphia.
Great. Thanks so much.
Thank you very much.